Start.
Hi again. Can you hear me?
Yes.
Okay.
Apurva, start.
Huh.
Good afternoon, everybody. First of all, on behalf of organizing committee, myself, Dr. Apurva Giri, coordinator, Department of Food Processing, and assistant professor and head, Department of Nutrition of Mugberia Gangadhar Mahavidyalaya, and convener of this webinar, convey my heartiest thanks to all of you for joining with us in this one-day international webinar on strategies to boost immunity by functional foods to combat COVID-19, organized by Department of Food Processing in collaboration with Department of Nutrition and Research Cell of Mugberia Gangadhar Mahavidyalaya. Respected Chairperson, Dr. Shapan Kumar Misra, Principal of Mugberia Gangadhar Mahavidyalaya, our all honorable speakers, Professor Anthony Gomes, ex-professor and UGC Emeritus, Department of Physiology, Calcutta University, India, Dr. N.M.N.K. Narayana, Senior Lecturer, Department of Animal Science, Faculty of Agriculture, University of Rihana, Sri Lanka, Dr. Subrata Hathi, Assistant Professor, Department of Dairy Microbiology, SMC College of Dairy Science, Anand Agriculture University, Dr. Marian Linior Silva, Assistant Professor, and Dr. Alexandra Bernardo, Associate Professor, Igas Moniz University, Portugal. Ms. Koel Pal Choudhury, Research Dietitian, SSKM Hospital, Joint Convener, Dr. Bidhan Chandra Samanta, Dr. Kalipada Maiti, Coordinator of IQAC, and the faculty members and students of this college, and all the participants from India and abroad. It is my pleasure to inform you that in this webinar, more than 1,200 participants registered. Dear participants, introducing this organizing institute, Mugberia Gangadhar Mahavidyalaya was established in culturally rich area Mugberia in 1964. The college is affiliated to Vidyasagar University. At present, the college is NAAC accredited with B+ grade in third cycle, having science, arts, commerce stream, community college, and Bivak degree program. The college is recognized with College with Potential for Excellence by UGC and Star College by DBT Government of India. In this college, nutrition department is started in 2006, and in 2018, to make skilled youth, the college opened Department of Food Processing to offer Bivak degree program in food processing with the approval and funding of UGC. Dear participants, coming to the theme of the webinar, friends, we are very much experienced that we are now in a critical situation of COVID-19 pandemic. Throughout the world, people are restless to find out the ways that can minimize the risk of COVID-19. I think healthy eating habit is one of the remedial ways for this. The people who are weak in immunity are prone to attack COVID-19. Functional foods are those foods which have health beneficial role beyond its basic nutrition. So, functional foods which can improve the immunity of the people is helpful to combat COVID-19. It has been scientifically reported that fermented dairy products, probiotics, prebiotics, omega-3 fatty acid, some medicinal plants like ashwagandha, giloy, tulsi, shatavari, can act as potential for immune boosting. In addition to food components like some vitamins like vitamin D, E, C, B6, B9, B12, and some minerals like zinc, selenium, magnesium, and some herbs, condiments, and spices like ginger, garlic, moringa, mint, cumin, cinnamon have immune enhancing role. The present international webinar will provide more information on this. Now, I would like to request our Honorable Principal of Mugberia Gangadhar Mahavidyalaya and Chairperson of this webinar, Dr. Shapan Kumar Misra, to inaugurate the webinar by his valuable inaugural address. Sir, please.
Good afternoon, everybody. We are grateful that we have 5 eminent personalities in this webinar. Respected Dr. Maria, Assistant Professor of Aegis Manis University. Good afternoon, everybody. And respected Dr. Alexandria, we have Associate Professors of Aegis Manis University from Portugal. Respected Dr. Narayanan, Senior Lecturer, Department of Animal Science, University of Rahuna from Sri Lanka. Respected Professor Anthony Gomes, University of Calcutta. Respected Dr. Subroto Hathi, Assistant Professor, Department of Dairy Microbiology, SMC, College of Dairy Science, Anand Agricultural University, Gujarat. Dr. Apurba Giri, Convener of the seminar and HOD, Department of Food Processing and Nutrition of the college. Dr. Bidhan Chandra Samanto, Joint Convener and HOD, Department of Chemistry of the college. My colleagues, researchers, students, and audience. Now it is my great opportunity to welcome all of you in this webinar on strategic to boost immunity by functional foods to combat COVID-19. Immunity power is an important mechanism of human body to combat external harmful virus, bacteria, fungi, different microbes, etc. To build our immune system effectively, we need to take food with proper recommended nutritional elements. We know the 6 basic nutritional elements are carbohydrate, protein, fat, vitamin, minerals, and water. All these elements should be in our diet with good proportionality. Out of good proportionality of these elements in our diet could create malnutrition, preventing from building proper immune system in the human body. Hence, we should take, apart from normal food like rice, bread, plus meat, we need to eat a lot of fresh fruits and vegetables, nuts, seeds, probiotics, etc. and drink sufficient amount of water. Boosting immune system is now especially very important issue during this COVID world. At present, about 13 million people are affected by novel coronavirus around the globes, and death tolls reaches to above 500,000. We will hear from the resource persons about the functional foods that could boost immunity for fighting COVID. However, I think we should maintain ICMR protocol like using masks, creating social distancing, washing our hands and mouth, and yoga activities for combating this pandemic. I do hope that this sort of interaction and exchange of news and views in a seminar will greatly enrich researchers and students, as well as this will be helpful to equip the people with proper implementation. It is my great pleasure that the food processing department of our college has been organizing this type of seminar in collaboration with our research cell and nutrition department. Last but not the least, I express my profound respect and best wishes to all respected guests and speakers for taking interest in this matter and giving us their valuable time. Thank you. Thanks, all of you.
Thank you, sir, for your valuable address in front of our distinguished participants. Now, I would like to request joint convener of this webinar, Dr. Bidhan Chandra Samanto, coordinator of research cell, Mugberia Gangadhar Mahavidyalaya, to say something about research cell and relevance of this webinar.
Thank you, Apurva. Good afternoon, everybody. Respected Chairperson, Dr. Swapan Kumar Mishra, Principal Mugberia Gangadhar Mahavidyalaya. Respected, honorable, distinguished guests and speakers, my dear colleagues, students, and dear participants. First of all, I would like to convey my heartiest thanks to all of you for joining to our international webinar on strategies to boost immunity by functional foods to combat COVID-19. Dear participants, it may be stated for your kind information that Mugberia Gangadhar Mahavidyalaya is one of the renowned institute of Purba Medinipur district, West Bengal, India, in respect to not only its academic activities, but also its research and social development activities in the nearby locality. Research cell is one of the potential wings of the college. The cell throughout the year encourages both faculty members and students to engage themselves in various research activities and hand-on experiments by using local resources. In the initiation of research cell, the college has set up vermicomposting production unit in the college campus to provide this composting manure to the local farmer at an affordable cost. Not only that, the research cell jointly with the Department of Botany of the college has created a herbal organic garden and developed a herbal product named Moringa Digestive from the Moringa leaf harvested in our garden, college garden. In the very recent, the research cell has organized a national webinar on development of new trends of scientific research to combat COVID-19 in collaboration with Environmental Epigenomics Lab, Department of Environmental Science, University of Calcutta. On the continuation of this type of activity, this is our another collaborative work. Dear participants, as you know that in the national lockdown period due to COVID-19 pandemic, the academic activities and research works have been hampered very badly, and we are passing through a very difficult time. But we must be optimistic that we will start our work again and engage our students in a very fruitful and constructive ways. Doctors, scientists, researchers throughout the world are trying their level best to find an effective treatment or effective solution by inventing medicine or vaccine, or by inventing detecting tools, or by ways to boost the immunity of us to reduce the risk factor of COVID-19. So we are very much eagering to hear from our speakers how functional foods can play an important role to boost the immunity of people to reduce the risk factor of fatal novel coronavirus. We should keep in mind that we will win the fight and will able to overcome the crisis created due to COVID-19. So I think I should conclude here. The session is very lengthy. So thank you. Thank you all again to joining with us. Thank you. Apurva.
Thank you, Dr. Samanta, for sharing your valuable views with us. Now, we will start technical session of this international webinar on strategies to boost immunity by functional foods to combat COVID-19. I'll request Dr. Bidhan Chandra Samanta, Associate Professor and Head of Department of Chemistry of Mugberia Gangadhar Mahavidyalaya to coordinate the technical session 1. Dr. Samanta, please.
Thank you, Apurva, for handovering the platform to conduct the technical session 1 as session coordinator. Thank you. As you know, dear participant, as you know, in this session, we have 3 speakers, Professor Anthony Gomes, Professor Dr. Narayana, Professor Subrata Hathi. So, according to the schedule, we have the first speaker, Professor Anthony Gomes. Before inviting him to his valuable lecture, let me give a brief introduction about Professor Gomes. Professor Anthony Gomes is ex-professor and UGC Emeritus UGC BSR Fellow of the Department of Philosophy, Calcutta University. He had his pre- and post-doctoral research training on venom and toxin at Calcutta School of Tropical Medicine. He worked at Tohoku University, Japan with INSA JSPS Postdoctoral Exchange Program Fellowships. He has published 145 scientific articles in peer-reviewed national and international journals. Professor Gomes has 2 national and 1 US patent, written 4 books chapter, guided 32 PhD students. He is fellow of several Indian societies or associations. Professor Gomes is a member scientist of the Task Force on Venoms and Toxins, ICMR Government of India, past founder General Secretary of Toxicological Society of India, founder President Association of Biomedical Sciences, Kolkata, India. He is a CPCSEA Government of India main nominee of several prestigious institutes of West Bengal and a member of CSC Government of West Bengal. With this brief introduction, I would like to invite Professor Dr. Anthony Gomes to deliver his valuable lecture on fight against COVID-19 with nutrition and dietary fiber. Sir, please start your lecture.
Hello?
Sir. Yes, sir.
Is there the slide?
No, sir, not visible. Yet down.
Not coming.
Please share your slide. Yes, sir, coming. Give it presentation mode, sir.
I can't see the slide.
Yes, visible, clearly visible.
But I can't see.
You can't see, sir.
No?
We are,
Yes,
we are
now?
yes, sir, visible.
Okay. Now it is visible.
Yes, sir. Please
Okay.
start your. Okay.
Good afternoon, everybody, and welcome participants and speakers and the staff of this college, the principal of this college. I would like to thank the principal of this college and the organizing committee to invite me to join this seminar. This slide I have made mostly for the UG and PG students. Before I go to my talk, I would like to tell you one small story. It was in the March 13th when I gave my first lecture on COVID at MBB College, Tripura on 13th March. And after that, I ran away from there to come to my home and I stayed locked down. Till then, this is my second lecture on COVID after a long gap. So thank you, sir, for inviting me to break my lockdown in lecture series. So my talk is fight against COVID-19 with nutrition and dietary fiber. These are the lecture outline, what I'll talk within 20 minutes. Objective, few words on COVID, very few, basic, dietary fiber, basic, how nutrition and dietary fiber will help us to fight against COVID, and finally summarize. The objective of this lecture to aware our next generation students and teachers how to survive against viral diseases by increasing body immunity with the help of nutrition and dietary fiber. WHO declared COVID-19 outbreak as a global emergency pandemic on 11th March 20, which originated from Wuhan city of China. Till today, no antidote, no vaccine exists, only symptomatic treatment. We are living in the age of bioterrorism using bioweapons. 2019, we have witnessed COVID-19, maybe 21, a new virus, and maybe 22, God knows, another virus. So it's a warning for you, be prepared for a long battle against viral diseases. The question, in India, total COVID-19 infected people are 906K, death 24,000, recovered 571. This is very alarming that nearly 65% recovered. My first question, without antidote and vaccine, how they have recovered? It's the herd immunity and the symptomatic treatment. My second question, how you can increase your herd immunity? Nutrition and dietary fiber, and especially the functional foods, may help you to increase your immunity to fight against COVID-19 virus. This is the focal theme of this webinar also. What is a virus? Immunological threats are initiated by antigen. They are foreign bodies, which may be protein, peptides, and many other things. Viruses are submicroscopic infectious agent found in every ecosystem, and they are capable of infecting living organisms, including humans. When a virus infects a host cell, the host cell gets enslaved to the virus and synthesizes thousands of identical copies of the original virus. Thereafter, the host cell dies and the process increases newly synthesized virus particles continues. This is the culprit. You can see diagrammatic structure, the genomic RNA. The virus is around 60 to 100 nanometer diameter, very small. But don't neglect this. So small is it has flattened Big B also. We have seen in that from the Bollywood star. I hope Big B will recover very soon. And this appears around round and oval, stable on plastic and stainless steel, inactivated by UV and heating. And recently, the US president, there are many nomenclature, but this is something funny. The US president on 21st June named this virus as Kung Flu virus. So this Kung Flu virus that breaks our body immunity. We'll see how it breaks. Before that, the clinical features, when it breaks, enters the body, these are the clinical features. There are fever, fatigue, decreased kidney function, there is diarrhea, circulatory problem, and this is the main effect on the respiratory system. These are the clinical signs: sneezing, runny nose, sore throat, dry cough, shortness of breathing, and this is mainly due to the many kinds of immunological consequences that happens in the body due to COVID. I'll see how COVID-19 that breaks our immune system. See, COVID-19 breaks our immune system through cytokine storm. See, you see after entry attachment, and ultimately it goes inside, and at the molecular level, it creates havoc, and thereby releasing cytokines and damage-associated molecular patterns. All these lead to the increased secretion of cellular secretion by the cells of huge amount of cytokines, what we call the cytokine storm. And this cytokine storm that leads to inflammation, immune coagulation dysfunction, and ultimately sepsis and multi-organ failure. So somehow we have to stop this cytokine storm. There are a lot of works going on throughout the world regarding the making of inhibitors of cytokine storm, but I'm not going in that. I'll come straight to the the diagnostic test and management. WHO recommends sample collection from the upper and lower respiratory tract. They are assessed by the RT-PCR. Isolation is the most effective measure, and the treatment is only symptomatic care and oxygen. Critical care patient, of course, need high flow oxygen and steroid therapy. Till a vaccine is available, improve your immunity. Improve your immunity. And this is the dietary fiber, what we are going to speak today. So what is dietary fiber? It has been defined as the plant polysaccharide, including cellulose, hemicellulose, pectin, lignin, which are not digested by the human digestive enzymes and so contribute to the fecal matter. You may ask me, what is the daily requirement? The American Diabetic Association, as well as American Cancer Association, they say that 40 grams per day per person of dietary fiber is required, which you may have from cereals, fruits, and nuts and vegetables. You see some unique figure I have given here, lady's finger. That's a wonderful vegetable having 6.2 grams of fiber per cup. The cereals, the guava, one of the very good fruit, 3.6%, coconut 7.6, cornflakes 11.5, and this is my favorite jhalmuri, which I used to take while I was teaching in Jhargram Raj College. While returning in the local trains, all of us used to take this jhalmuri. It's a unique combination of lot of fibers. You can see nuts, chili, carrot, lemon, chana, and lot of things. So take on jhalmuri. So good and bad effects on dietary fiber. The good effects are they reduce appetite, increase satiety, decrease energy intake, alter gastric emptying, lower blood cholesterol and glucose, alter fat absorption, and decrease transit time. It also increases fecal weight by water holding hypothesis. They reduce constipation, reduce gallstone formation. Among the bad effects, remember that it has both good and bad effects. So the correct proportion is very important. It may alter vitamin and mineral and drug effect, alters microflora, may produce pro-oxidant and colon cancer. It produces GI tract irritation, flatulence, and intestinal obstructions also. Nutrition status and role of diet, they are the key in the lifestyle, what we have to maintain, especially in the COVID-19 infection. A healthy balanced diet can offer the necessary macro and micronutrients, prebiotics, probiotics, and symbiotics. They are having all sorts of anti-inflammatory immunomodulatory activity, and that is required especially for the elderly and the young. They can restore and maintain immune cell function, thus increasing protection against COVID-19. The cereals, fruits, and vegetables having antioxidant, vitamins and minerals, and phytochemicals show anti-inflammatory, antioxidant effects against COVID-19. But we need more research data to establish the specific protective role of dietary fiber against COVID-19. Fish oil, marine products like alga and seaweeds. In Bengali, we call it tok palung. You know, the seaweeds are very common nowadays. People are taking a lot of seaweeds in this area, but it's mostly taken in China, Japan, and that side is, and it has been associated with various health effects, including cardiovascular disease and cancer. Especially the PUFA has been shown to exert anti-inflammatory effects through PG synthesis. Marine fish intake, at least 2 to 4 serving portion per week, that will decrease the risk of reduction against viral infection. But it is not clear that fish and fish oil consumption may be beneficial against COVID-19. We don't have much data. What is required? More detailed studies. Vitamin C, already the principals have told us that many vitamins and minerals that are helpful, like vitamin C, ascorbic acid, is a water-soluble vitamin, and which is very commonly used in cold and flu conditions. Nobel laureate winner Linus Pauling, 1970, he first established this fact, and later it was established that it helps in cold and flu. The recommended daily allowance of vitamin C, 100 milligram per day. Some of the rich source in our country, amla, lemon, chili, guava. Although evidence is currently weak in relation to the utility of vitamin C against COVID-19 infection is very weak. Vitamin D, increasing vitamin D, the sunshine vitamin intake may reduce risk of viral infection. Vitamin D antiviral effects by interfering with viral replication and through its immunomodulatory and anti-inflammatory properties. Low vitamin D level may play a role in high incidence of COVID-19. The recommended daily allowance is 600 to 800 international units per day. Rich source of vitamin D are egg, mushroom, fatty fish, milk, and dairy products, and foods fortified with vitamin D. I will ask the students to remember these things, vitamin D sources, but more research required, before we confirm the prophylactic and therapeutic values of vitamin D against COVID. Vitamin E, another very important vitamin, alpha tocopherol, is a group of fat-soluble antioxidant vitamin. It may exert in immunomodulatory enhancing effects by scavenging oxygen species to reduce oxidative stress, regulate the production of ROS, reactive nitrogen species, and modulate signal transduction. The rich sources: nuts, seeds, green leafy vegetables, fortified cereals, vegetable oil. The recommended daily allowance is 15 milligram per day, according to NIH USA. However, there is little evidence that till today, the utility of this vitamin as a prophylactic or therapeutic agent against COVID. It has been suggested that combination of vitamin C and E may be useful as an antioxidant for cardiac complication of COVID-19. That is paper, they have said that both these vitamins may be useful. Among the minerals, there are many minerals, as earlier principals said already told. I'll speak on only two minerals. Zinc is a trace element that is critical to the development of immune cells and an important cofactor for many enzymes. The common sources are meat, dairy, legumes, nuts. The rich source are lentil, rajma, soybean, say 3 to 5 milligram percent. Bengal gram, very good source, 6 milligram percent. Coconut, 5 milligram percent. Groundnut, 12 milligram percent. The recommended daily allowance is 8 to 11 milligram per day. The zinc intake at this level, 30 to 50, that might aid in the control of RNA virus such as influenza and corona. Copper is also essential trace element found in cereals, fruits, and nuts. Recommended daily allowance 30 milligram per day. A copper intake of 7.8 milligram per day has been shown to reduce oxidative stress and alter immune function. To summarize, increasing the consumption of both soluble and insoluble fiber through the following vegetables and peas, nuts, lentils, fruits, etc., that will be helpful against viral attack. That is definitely. It is important that this is another important issue, that pseudo-scientific and false claim associated with functional food are to be avoided. In this pandemic, businessman people, they are confusing us. So you have to be very careful about the pseudo-scientific and false claim about functional foods. To conclude, it is vitally important to maintain a healthy diet and lifestyle during the pandemic. Extensive research is necessary, and this sort of awareness program should be done not only in college, but at the school level also. More programs should be there. These are the references for the students. Acknowledgement. Thank you again, once again, Principal, and I thank especially Dr. Apurva Giri. Give a hand for Apurva Giri, please. And please follow me. We can beat it definitely together. Thank you very much, and God bless you. Thank you.
Thank you, sir, for your very much informative talk. And we have received a lot of appreciation for your talk.
Thank you.
And we also thank you for your suggestions. We must keep in mind to organize such type of awareness camp in school levels also. Participants, those who are wanting to ask questions in this lecture, please write your question in YouTube chat, mentioning your name and name of the institute. We will describe all those questions in the question answer session. So we are requesting participants, those who are wanting to ask questions in every lecture, please write your question in YouTube chat box, mentioning your name and institute. We will describe these questions in the question answering session. Thank you, sir, for sparing your valuable times with us. We will again invite you in the question answer session.
Okay.
Thank you, sir. Our next speaker in this session is Dr. Narayana. Before invite him, before invite her to her talk, let me give a brief introduction about Dr. Narayana. Dr. Mrs. A.N.M. N.K. Narayana is a senior lecturer of Department of Animal Science, Faculty of Agriculture, University of Ruhana, Sri Lanka. She completed her BSc in Agriculture Honours and MPhil from University of Paradenia, Sri Lanka. She obtained her PhD from National Dairy Research Institute, Karnal, India, in the field of dairy technology. She attended international training programs on dairy farming, milk processing, molecular biology, and biotechnology techniques offered under different fellowships from different countries. She has over 40 research publications in national and international journals and conference proceedings. Her main research interests are functional dairy products development and sensory evaluation techniques and also consumer studies. Now I would like to invite Dr. Mrs. A.N.M. N.K. Narayana to deliver her talk on potential applications of immune boosting medicinal plants in functional foods to combat COVID-19. Madam, please start your lecture.
Yeah, thank you very much. So,
Madam, please share your slides. Okay.
thank you very much for inviting this international webinar. So, today I am going to present my presentation on potential applications of immune boosting medicinal plants in functional foods to combat COVID-19. So, all of us know that the immunity is the balanced state of having adequate biological defenses to fight infections, diseases, or other unwanted biological invasions while having tolerance to avoid allergy and autoimmune diseases. So, these immune responses are the result of an effective interaction between innate and acquired immune systems. So when pathogen invades the body, the innate immune system activates first, and then the uh, dendritic cells in the innate immune system will transmit the pathogen information to the adaptive immunity system. Then adaptive immunity system will activate and it will produce killer T cells and antibodies and specifically attack the pathogen. So for our immunity, this innate immune system and the adaptive immunity both are very important. So as you all know, COVID-19 infection is known to affect individuals with weak immunity more severely. Now, because of that, more than ever before, it is important to treat our body well so that our immune system is geared up to handle viral infections. Because SARS-CoV-2 doesn't have any known cure or treatment. So that is why it is very important. But many factors play a significant role in altering the immunocompetence of a person. For an example, age, sex, genetic variability, stressors, alcohol, drug abuse, like conditions, including malnutrition, especially environmental pollution, lifestyle. Those things alter the immunocompetence of a person, and because of that, it may affect susceptibility to and severity of infection. So, importance of diet on immune health is becoming increasingly well recognized. Now, because of that, there is a massive opportunity for functional food and drink market. Functional foods are emerging as the best strategy to tackle nutrition-related immune system compromises. Actually, all the foods are functional because they provide taste, aroma, nutritive value, and because of that, all foods can be considered as functional. But especially, functional foods are whole, fortified, enriched, or enhanced foods that provide health benefits beyond the provision of nutrition, essential nutrients. So these are some of these are the categories of functional foods and some of the examples of the functional foods. Conventional foods such as garlic, nuts, tomatoes, then animal source foods like milk, meat, those are coming under conventional foods. And we can develop functional foods as well, like modified foods, fortified, enriched, enhanced foods, some medical foods, and foods for special dietary users like infant foods are also comes under functional foods. Even though these functional foods are healthy, consumers want taste because from previous findings, it was found out that this buying behavior consumers of foods and any item is influenced by various motives. So even though the functional foods are healthy, if they are not tasty, people are reluctant to buy those foods. So because of that, when we develop functional foods, we have to develop them with excellent sensory characteristics. So the functional food development, this is the success cycle of the functional food development. It has different stages from the conception of the idea, product development, and the major thing is we have to after developing a marketable product, we have to test the efficacy and tasty also, safety also, efficacy and safety, and using the evidences of those, we can have some publications and depending on the consumer, so the publications can be we can convey the message to the consumers, and consumers buying perception and interest is also very important for the success of a functional food in the market. So at present, herbs and spices, especially the medicinal herbs, are used to increase the acceptability of foodstuffs and improve the health. So the medicinal herbs are beneficial because they have different properties such as anti-mutagenic, anti-inflammatory, antioxidative, immune modulatory properties are there in medicinal herbs and plants. So according to WHO definition, medicinal plants are plants that contain properties or compounds that can be used for the therapeutic purposes or those that synthesize metabolites to produce useful drugs. So these medicinal herbs or the medicinal plants contain phytochemicals. Those phytochemicals are naturally occurring compounds with bioactive potential, some of which have potential immunostimulating activity. So these may act, the phytochemicals act individually or additively or in synergy to improve one's health. So these are some of the phytochemicals. I have listed here some phytochemicals present in those plants which are having properties. Yeah, which improve or boost the immune person. So now you maybe you have heard that this SARS-CoV-2 virus, this ACE2 receptor for the SARS-CoV-2 virus. So after that, it will invade the body and create those disease conditions. So it will inhibit the normal, block the normal function of the ACE2 also after viral replication. So these medicinal plants, it is found that some of the medicinal plants, some bioactive peptides or the phytochemicals in these medicinal plants will inhibit will inhibit the viral replication and also by inhibiting viral polymerase. Also some chemicals and some plant compounds, they will inhibit the viral binding and entry to the body. They can inhibit the infection and further conditions in the body. So this is, it was shown here, this Otunia core data, that fish mint, that plant is available in Southeast Asia. So it is normally used as a leafy vegetable and cooked with other vegetables, fish, meat, because it has a fishy mint also. So we can use this, actually, there is a potential to be used this fish mint in functional fish and meat product developments. And this sweet wormwood, it is it has a lot of antioxidants, and because of that we can use this as a food antioxidant in real food systems instead of having some BHA like synthetic antioxidants. So it will have so this is Lycoris radiata. It also has some effect on the pathogen entries. So the colloidal extracts from the bulbs have a possibility to be used in food industry to replace gum arabic. And however, the starch from the bulbs are also used in the food, but there are some poisonous materials also. So we have to remove them by bleaching the food before cooking. And this is Japanese nutmeg. So the seeds, raw and cooked, is normally used in confectionery items. And this also has a potential to be used in some functional foods because it has some effect against SARS viruses. It has found that because of due to the flavonoids, it has some effect on the viruses. And this black cumin, it is actually the spice. It is normally used in food systems. You can see here it has used in developing cheese, or actually we can just add black cumin because it also has some effect against these viruses because they will inhibit, they are having some the inhibition activity due to the phytochemicals present in them to against COVID-19, COVID virus. And another promising herb is ginger. Actually, ginger contains phenolic and terpene compounds. This ginger, normally it has already used in many functional foods. And it also has a promising effect in some against these viruses. So there are a lot of research papers found that the sensory properties of ginger, spice, yogurt, and cookies also. And in the current market also commercially, lot of products are available that was made using ginger also as an additive. So it is a promising herb which can be used in further functional food developments. And this is Withania somnifera. It is called as Indian ginseng. Actually, this ginseng, from in silico studies, it has been shown very recently. It has been found that this vithanone in this ginseng has a distinct effect in the interface of ACE2 RBD complex. So it will block and weaken the entry of the virus into the body. So because of that, this ginseng has a very high potential to be used in functional food applications. Actually, this it is local name is called as ashwagandha. In Sri Lanka, also we called ashwagandha. This Withania somnifera is used in many functional food applications. So there are so many findings, literature findings. So we can develop further this very valuable medicinal herb into some acceptable food sources. Another important one is Thinosphoria that is giloy. In Sinhala, it is called as rasakinda. So this rasakinda is also having very high potential to be developed into functional foods because in this recent study, they have mentioned that it also, this tinagod site found in this giloy, uh, there is an effect in this complex. So it will inhibit this RBD is complex forming that the receptor this binding ability. So giloy would be a viable option to control SARS-CoV-2 entry into host cells. So, and also the general immunomodulatory nature of giloy due to some different phytochemicals would enhance innate immunity also against virus entry. And another important herb is this tulsi powder is also commercially available, Osimum sanctum. It also have shown that tulsi phytochemicals, so many phytochemicals are there, including this phytochemical, which is having some effect to inhibit the coronavirus replication and growth and spread. So Tulsi incorporated products are also available commercially, and going on. Tulsi powder is also available commercially, and so many opportunities are there to the develop this Tulsi powder. And another very famous herb is garlic. So there are different products are available. The functional foods are available. So these polyphenols and organosulfur compounds are the major compounds in garlic. So we can develop so many functional foods to have a beneficial health effects, even though we actually this garlic is normally used in everyday food applications as well. And the aloe vera is another promising financial products are also available: yogurts, ice creams, then yogurt drinks, dairy snacks. So many things are available, and there is a huge potential to be used in many functional food applications in the future also, because it contains a lot of antioxidants and vitamins, enzymes, minerals. So many things are there which can fight against the viruses and bacteria like diseases by boosting our immunity. And fenugreek, this is incorporated cheese. Fenugreek also have flavonoids, different chemicals, phytochemicals, and fenugreek also have a potential the application in the functional foods to boost our immunity. Then this thyme tea also has been found to inhibit the entry of viruses to the body because of the special phytochemicals containing in them. So it is available as a herbal drink. and these also can be used to boost our immunity. And some other this herb uh, and chamomile tea also we can use because of these special phytochemicals present in them to boost our immune system and to avoid uh, the entry of viruses to our body, invading the viruses to our body. This is a spice actually, Salvia officinalis. There are some phytochemicals in it which can boost our immune system, and these are used to make actually to the day-to-day food preparations also. It is used as a spice, and also this some special kind of cheese varieties it is also used. It is very popular in some European countries also. This another herb, rosemary. It also used as a seasoning in variety of dishes, and this due to some phytochemicals present in this, it is also used. It can be used in immune boosting food applications. And cinnamon is very common spice in Asia. So this normally in day-to-day cooking also we use, and a lot of research findings are there to apply in functional food preparations, dairy products, and also in some other foods as well. So it is also a very good immune boosting spice which can be used in functional food preparations. So there are so many other the herbal products as well, but those are specifically I mentioned because they have a lot of phytochemicals and a lot of food applications, because consumers normally they need to have the tasty food also. So we have to think about those things as well when we develop functional foods. So I think there is a good future for functional food development with these medicinal herbs, and they can be used to boost our immune system and to avoid the disease incidences. So with this, being with us.
Thank you, madam, for your very much enriched and informative talk. Really appreciate. We really appreciate your talk. I think so much question from audience also. These will be described in our question answer session. So we will again invite you in the question answer session. Thank you, ma'am, for your talk and sparing time for us. Thank you, ma'am. In this session, our next speaker is Dr. Subroto Hathi. Before inviting him to his talk, let me give a brief introduction about Dr. Subroto Hathi. Dr. Hathi at present is working as assistant professor in the Department of Dairy Microbiology, SMC College of Dairy Science, Anand Agriculture University, Anand, Gujarat, India. Dr. Hathi published more than 55 research papers in reputed journals nationally and internationally, 8 review papers and 3 edited books. He is the secretary of Southeast South Asian Network Fermented Foods, Anand, and executive editor of International Journal of Fermented Foods. He is also the editorial board member of Letters in Proteins and Peptides. He also handled the extremely funded projects sponsored by DST, DBT, ICAR, Government of Gujarat as PI or co-PI. Dr. Hathi is a recipient of numerous prestigious awards, which include NAAS Young Scientist Award sponsored by NAS, New Delhi, Professor Sukumar De Gold Medal from WBUAFS, Kolkata, as university topper, Urmila Bala Gold Medal from Indian Dairy Association, New Delhi, Early Career Research Award by DST, New Delhi, IACBG Fellow by DBT, New Delhi, INSA Visiting Science Priest Award 2018 by INSA, New Delhi, Best Teacher Award 2018 by ICAR and AAU, Anand, and also recipient of Dr. Gauri Ganguly Memorial Award 2019 by Indian Science Congress Association, Kolkata. He visited the University of Copenhagen, Denmark for presenting the research work under Indo-Danish collaborative workshop in 2018. With this brief introduction, I would like to request Dr. Subrata Hathi to deliver his lecture on application of probiotics in immunity boosting. Sir, please start your lecture.
Good evening to all. I think you are watching my first slide and my voice also. It is okay
Yes,
now?
yes, sir.
Okay.
Okay, sir.
So very good evening to all of you. First of all, I would like to give thanks to Dr. Apurva Giri because he invited me to speak on a very relevant topic nowadays. This is application of probiotics in immunity boostings. So before going to the topic, I want to give a brief introduction on it. You may see my first title slide that it has been written that a billion bacteria a day keeps the doctor away. Means if you are taking a bacteria means good friendly bacteria, those are probiotics, we basically tell them, or we basically termed as probiotics in our language. That bacteria keeps your intestinal gut system very healthy. So these bacteria will keep you very healthy, and 30 more than 70% of your immunity depends coming from your intestine. So that will maintain your health also. That's why I have written a billion bacteria a day keeps the doctor away. Just like if you eat apple a day, you have to be away from the doctors. So first, coming to the very basic idea about the probiotics. You know, the probiotics was first observed by the Russian Nobel Prize winner, Dr. Eli Mechnikov. And he actually observed that some acid producing bacteria in the fermented milk products, they are giving some extra benefits to the Bulgarian peoples, and they are living longer than the other communities. And from that, then he observed that what kind of foods they used to consume, and he find out that some good bacteria there who are giving them this kind of extra benefits. So first he has given the concept of probiotics, and later on in 1930s, Dr. Minoru Shirota, he is from Japan, and he developed a fermented milk product. All of you know it is a Yakult, and it is available all over the world now. And in our country, even you can get it from any retailer shops. And he used only one bacteria, that is Lactobacillus casei shirota, and that bacteria is doing wonder. And he has first commercialized this probiotic drink, and most of the peoples they have accepted it. In 1965, Lilly and Stilwell first give the term probiotics. They have given the term probiotics. So if you look back the history of the probiotics, it was first started on 1857 when Louis Pasteur, he first said that the lactobacillus, they are giving some health beneficial properties in the intestine. And then 1878, Joseph Lister, he isolated the lactic acid bacteria, means the lactobacillus. And in 1899, Henry Teaser, he isolated the bifidobacteria. This is another very potent bacteria coming under the Firmicutes family. And in 1905, Eli Mechnikov, which I have just told you the actual concept that yes, these bacteria have some benefits and we are getting out of it. And 1930, I have already told that the Dr. Minoru Shirota, he first commercialized the bacteria in the form of a very good product and which is acceptable all over the world. So before going to the depth of the probiotic and their different benefits, first we see the definition of the probiotic. FAO and WHO in 2011, they have given the complete definition of probiotics, that live microorganisms that when administered in adequate amounts, they confer a health benefit on the host. But ISAP, Indian Society for Probiotics and Prebiotics, they changed the word. Just you may observe my slide that at the end it is written live microorganisms which when administered. So they have just changed a minor grammatical changes of this paragraph and make the complete sense of the definitions. So what they did, they said you just change which with that, and then you will get a very good complete definition of the probiotics. So now that definition is accepted all over the world. It is just a minor grammatical change given by the ISAP in 2014, and it is also published in a very good journal. Now the question is that why we need the probiotics, and why the probiotics is getting so much importance nowadays. So what are the basically factors are affecting the intestinal micro ecosystems? All of you know that when you will go to the doctors for any kind of sufferings like flu or other kind of fevers, they used to prescribe the antibiotics or other drugs, and that antibiotics goes into the intestine and they disturb your native microflora. So when your native microflora will get disturbed, then you can't get the immunity or your total digestion system, everything will get changed. And if you have some microbial infection like diarrhea, it is caused by salmonella or Shigella or Clostridium difficile. So these kind of organisms, they basically what they do, they mostly they change the microbial systems, and secondly, you can see that these microbial infections totally disrupt the your native microflora. And if you are taking the diet, highly processed diet, and that diet doesn't have the fiber, means low fiber foods, that also affect the natural microflora. Secondly is the chronic diarrhea. Just I told you that Clostridium difficile or Salmonella or Shigella, they are also causing the diarrheal very the diarrhea which mostly good bacteria are getting killed or suppressed, and their number is increased. And even if you are suffering from some kind of stress and lifestyle diseases, the natural flora, they are also getting disturbed. And these are the factors are very much important for the probiotics who can heal these kind of problems when you will take or probiotics from the food, through the foods or as a supplements. I have just come across that in 2018, 4,900 papers have been published on gut microbiota and probiotics. So you see the scope of the probiotics. Lot of people are working till date, and they are publishing lot of works on probiotics and their different aspects of the human health. And mostly in human, I think 100 trillion bacteria are present in your intestine, maybe a good or bad. So when you will see the bacteria means probiotics, how will you select them? How will you select these organisms? First, you have to go for safety test. Safety test means the bacteria should be safe. So the origin of the bacteria is very important, from where you are isolating the bacteria. And secondly, safety means the bacteria should not contain any pathogenicity, any disease producing genes should not be there, which we will call it a pathogeny islands. And functional aspects mean the bacteria should not have any kind of functional aspect means the bacteria should produce lot of acidity when you will prepare fermented milks, and they should be stable during the processing of the foods. And another important point is survival. Survival means when they will pass through the GI tract, they should be active, physiologically active. They should not get die. And another one, that adherence and colonization. When the bacteria will reach to the intestine, they should produce some mucin binding proteins which will help them to colonize inside the intestine, and they will get adhere. After adherence, they will grow and produce the different metabolites like antimicrobial products or antimicrobial substances like bacteriocins, peptides, or other organic acids. And then these bacteria will also help you to initiate your innate and adaptive immunity. And then genetic stability. All of you know about the antibiotic associated diarrhea or antibiotic resistance. So when your bacteria, live lactic acid bacteria, nowadays showing some antibiotic resistance pattern, because they have acquired the resistance antibiotic genes from the other bacteria through horizontal gene transfer, and that gene, that genetic stability should be there, so they should not contain or they should not accept the other genetic material from the other organisms. Another is the prevention of pathogens. The bacteria should not contain any kind of pathogenic genes or pathogenic islands, so the bacteria should be safe. And when you process the foods, you are doing the transportation or storage of the products. Throughout the storage and transportation, the bacteria should be active up to the end users. Now, in our country, ICMR, the ICMR, Indian Council of Medical Research, and DBT, both these organization has given a very good, the identification or evaluation of a probiotic systems. They have given a very good flow chart. First, they have given the strain identification. So you have to first identify the strain at the level of genus and species. So first is your target is to isolate and specify, then functional characteristics in vitro test. Then you have to go for safety assessment, efficacy assessment, and effectiveness of the assessment by doing the phase 1, phase 2, and phase 3 studies. Basically, these three are the human studies. So after getting success on the in vitro test or on animal study, then you can go for the phase 1, phase 2, and phase 3. Phase 1 basically safety assessment on the human trial, and efficacy means you have to keep one control and compare what kind of health claims you are targeting from your bacteria. And third one, you can take another control which is basically used for a maybe it's a drug, or which can be standard drug or standard treatment procedure, and compare your bacteria. So you have to go one by one through this prescribed format given by the ICMR DBT on 2011, and then only you can say that it is your probiotics. US FDA already recommended some probiotic strain and their sources, like Lactobacillus acidophilus, Bifidobacterium infantis, Lactobacillus fermentum, Lactobacillus rhamnosus, Lactobacillus acidophilus, Lactobacillus casei sirota, and then Lactobacillus reuteri, Lactobacillus rhamnosus, Lactobacillus johnson. And most of the companies, Danisco, Christian Hansen, Yakult, Danone, all over the world, they are selling these bacteria as a probiotic strain. Now coming to the COVID, because we are passing through a pandemic, COVID-19, and my topic is quite relevant to this immunity boosting. So some of the studies are being cited here, which are maybe it doesn't have a direct evidence, or you can say direct link to the management of the COVID, but we can indirectly say that the immunity system will help you a lot for fighting against these viruses. A Cochrane review of 12 studies involving 3,720 participants showed that the probiotics can help in preventing upper respiratory tract infections in all age groups. So here what they did, they have given probiotics, and they found mostly you can see your coronavirus, this also targeting the upper respiratory tract. So the bacteria, probiotic bacteria, also help to prevent the upper respiratory tract infections. So here they have reported. Another study, the probiotics has the power to enhance the immunity against the viral infections, with a special emphasis on the COVID-19, where they started the 43 studies, and they showed that the role of nutrients, probiotics, and enhancing the immunity. So in nutrients plus probiotics in combined, they are also helping to enhance the immunity. And the study, they said the daily intake of fermented milk with Lactobacillus casei strain sirota, they reduces the incidence and duration of the upper respiratory tract infections in healthy middle-aged office workers. So they have taken another clinical trial on Yakult culture. This is basically the culture is being for the production of Yakult, and they said that that culture also have the ability to prevent the upper respiratory tract infections. Another study that does not directly correlated to the probiotic, but fermented milk drink also have the ability to prevent the fever duration and detection by continuous taking a fermented milk drink. So these are the studies are being done for the control or the management of the viruses. All of you know that this coronavirus, this is basically evolved from the China in Wuhan city. So the Chinese scientists, they said that they have done one study and it is being published. So what they very recently they have published, they said that they have given probiotics to the COVID-19 patients in China, and they because they have seen that the persons who are suffering from coronaviruses, their gut systems, means the gut flora is getting disturbed, means microbial dysbiosis over there. So to prevent these secondary gut dysfunctions, they have given probiotics. And China's National Health Commission and National Administration of Traditional Chinese Medicine, they recommended the probiotics for the COVID-19 patients, even though there is no clinical evidence at all. But they said that because you see, when the persons are suffering from coronaviruses, the doctors generally giving the antibiotics, and I have already told you that the antibiotics are killing good or bad bacteria in your intestines, smaller part of the intestine. So your native flora are getting disturbed, means dysbiosis is there. So to prevent the dysbiosis and to restore your natural flora, they are supplementing the probiotics. And according to them, that probiotics may be used to maintain the balance of the intestinal microflora and prevent the secondary bacterial infections. This means that the foods and food ingredients that contain the probiotic particular or healthy bacteria in general may be consumed through the foods. Now come back to the immunity part, how your probiotics are giving immunity out of other benefits. So when you will consume foods, the foods will pass through your GI tract. GI tract has the largest surface area from where food is absorbed, and also the mucosal immunity is also produced. And the gut microbiota regulates both innate and adaptive immune homeostasis. I will discuss later on that what is innate and what is adaptive and how the bacteria is controlling them. So you come to the bacterial action on the immunomodulatory effect in the intestine. The figure, if you see closely, that this is the intestinal epithelial cell lines. The upper part is lumen. When the probiotics come, they enter through the TLR, toll-like receptors, and they enter inside the intestinal epithelium. This is called Proparia or Peyer's patches. So when the bacteria will enter inside the Peyer patches, they induce the dendritic cells, and as the dendritic cells will get activated, they produce different anti-inflammatory cytokines. So it is the adaptive immunity after probiotics initiating this kind of production of anti-inflammatory substances like cytokines, IL-10, IL, and T helper cells. They also produced. And when the virus directly infected the epithelial cells or M cells, then your macrophages or natural killer cells, they produce different anti-inflammatory substances, and they restrict the viral infection. So it is an innate response. So what is innate and adaptive immunity? We have just seen the figures. In the human gut, M cells present in the Peyer's patches are crucial. They have very crucial roles because they act as a transporter. They transport the macromolecules, antigens, microorganisms, and inert particles from the lumen into the lymphoid tissue by the adsorptive endocytosis. So basically, the different foreign bodies, when they comes very close to the lumen of the epithelial cells, the M cells, they transport these molecules to the lymphoid tissue by the adsorptive endocytosis. So when the antigenic molecule cross the intestinal barrier, they stimulate the innate as well as the adaptive immune systems in our body. The innate immunity is recognized as the first line's defense mechanism against the pathogen and can remember previous encounters while attacking them. Mostly in innate immunity, the phagocytic cells, including neutrophils, monocytes, macrophages, and natural killer cells, they are enabled as a first line defense system, and they act against the pathogenic microorganisms in the human body, which destroys the pathogens and protects from the corresponding infections. But the phagocytic cells and the proteins, which then activate the adaptive immune response. So you see here, the first line's defense mechanism is controlled by the innate immunity, as your phagocytic cells will produce different neutrophils, monocytes, macrophages, and NK cells. So after that, these phagocytic cells and some of the proteins which activate the adaptive immune response. So your adaptive immune response will act after the innate immunity. So it is a slower process compared to the innate immunity. And they are activated through the APC, antigen presenting cells. So if we talk about the adaptive immunity, the lymphocytes are the key players in the adaptive immune response, and they are more effective immune response, and they are having specific antigen receptors. This is called B cell receptor and T cell receptor. It is called B cell or T cell. So these cells contribute to adaptive immunity through the secretion of the antibodies. This is known as humoral immunity, while T cells contribute to cell-mediated immunity through the subdivision into T helper cell. This is mostly CD4 plus and CD4 plus we call it T helper cells, and CD8 it is called cytotoxic T cells. And B cells, they basically recognize the specific antigens through their B cell receptors, whereas CD8 cells recognize antigen as a peptide and measure histocomplex class 1 complexes, histocompatibility class 1 complexes, and CD4 cells recognize the antigen as a peptide, measure histocompatibility complex class 2. So this is their mechanism to recognize the antigen. So they are very specific against their antigens. Once the ACP are activated, T cells proliferate and differentiate into the CD8 cell and CD4 cells. And furthermore, CD8 cells convert into cytotoxic T lymphocytes, whereas CD4 cells, they activate and regulate the macrophages, and B cell respond to the adaptive immunity. So mostly your adaptive immunity is recognized by the B cell and T cells, and they are differentiated into the different cellular mediated immunity. Now, how the probiotics control this immunity in the intestinal epithelial cell lines. Basically, they produce the IgA secretion, means they not bacteria is not producing, they initiate the production of IgA, then production of cytokines, production of antibacterial substances, and they also enhance the tight junction of the intestinal barrier against the intracellular bacterial invasion, that competition with new pathogenic microorganisms for enterocyte adherence. And probiotic antigenic fragments such as cell wall compounds have the ability to cross the intestinal epithelial cells and M cells in the pairs patches, and then they also initiate the innate and immunity response in our systems. The immunomodulatory effect of the probiotic is attributed because of the release of cytokines, basically interleukins, tumor necrosis factors, interferons, transforming growth factors, and chemokines from the immune cells like lymphocytes, granulocytes, macrophages, mast cells, epithelial cells, and dendritic cells. So you see that the probiotics are initiating the innate and adaptive immunity. The cellular components, because all mostly the lactobacillus and bifidobacteria, they are gram-positive bacteria, and all the gram-positive bacteria, they have a lipoteichoic acids, and they stimulate the NO synthase enzymes, which is a potential in pathogen infection cell death mechanism. So they these lipoteichoic acids stimulate the NO systems. In addition, two surface phagocytosis receptor, one is the Fc gamma R3 and toll-like receptor, are also upregulated by the NO synthesis. So the NO, if the NO systems will get activated, then these are the anti-inflammatory substances or the immunomodulatory substances are produced, and they killing the pathogens. Probiotics also have been reported to interact with enterocytes and dendritic T helper and T helper 1 and 2 cells, T regulatory cells in the intestine, and also to modulate the adaptive immunity into the pro and anti-inflammatory actions. Oral administration of Lactobacillus casei, they also favored the rapid activation of the immune cells and produced a high number of specific markers such as CD206 and TLR2 cells. And TLRs improve the immunological defense mechanism in terms of pro and anti-inflammatory cytokine production upon the detection of foreign object. So here your probiotics, maybe a Lactobacillus casei, maybe a bifidobacteria, they initiate the adaptive immune systems through the cellular mediation and produce a lot of immunomodulatory compounds which basically kill the pathogenic organisms like Salmonella or other any viral infections. So here I will show that how the probiotics sensing the epithelial cell line. The first figure you can see here, the bacteria produce different metabolites like bacteriocins or different peptides or organic acids, short chain fatty acids. So they will get sensed through the TLR, toll-like receptors. They will transport it inside the M cells of the epithelial cell line. So they get transported. When they get transported, the BFF means B cell activating factor, and April, a proliferation inducing ligand. These two cells, BFF and April, promote the T cell dependent and T cell independent IgA class switching. So what the BFF will do, they produce the IgA. They are not directly producing, they are initiating the plasma cell to produce it. As the IgA antibody is produced, that will get transported outside the epithelial cell. These IgA molecules are transported outside by transcytolized by polymeric IgA receptors and retain the bacteria in the mucus. So in this way, your epithelial cell and cell is giving response to, means that is the crosstalk between bacteria and the epithelial cell lines. Now coming to the how the probiotics are maintaining the gut health. You see the second figure here, the gut lumen and then mucus. So in gut lumen, mostly all the bacteria are retaining. We have said the probiotic as a peacekeeping bacteria, means good bacteria, and some pathobiont or pathogens are also there. So when your peacekeeping bacteria is quite larger in numbers, your pathobiont cannot do any damage to the epithelial cell line. So it is a healthy gut. But if you are treated with some antibiotics or your diet is highly processed, low fiber content, and you are passed through some stress, or you are attacked by some viruses or pathogens, then your native flora is getting disturbed, means dysbiosis was there. If the dysbiosis was there, the peacekeeping bacteria number is getting down, and then your pathobiont was taking the or occupying the entire place of the intestines, and they are directly coming inside the Peyer's patches of the epithelial cell, and they ultimately they are entering into the circulation, means they are entering into the blood, and they are travel throughout your body, means they are reaching every organ to the human cell. One study, it is a very recent study, I have just come across, that they have said that the probiotic have a anti-tumor immune responses. It is the tumor has been, it is basically a mice model study. What they did, they have taken Bifidobacterium bifidum, and that Bifidobacterium bifidum they have injected into the mice in 100 microliter, and they are containing 10 to the power 8 cells, and they given for 20 days. And after inducing the tumor with this virus, human papilloma virus, and they have found that these are the immunoresponding or anti-inflammatory substances like IL-12, IFN gamma, IL-4, IL-6, and IL-10. But they have given two different treatments. One is the oral delivery of the Bifidobacterium to the mice, and another one is the intravenous injection of the Bifidobacterium to the mice. Only for IL-12 or IFN gamma, here you can see that the Bifidobacterium which are being administered through the intravenously, they are giving a good amount of IFN gamma, but otherwise oral delivery system was at par, and IL-12, if you see, it is significantly higher. So oral delivery of the Bifidobacterium bifidum and these kind of anti-inflammatory substances are producing in higher amount, and they are preventing the tumor proliferation in the mice model. Another is the effective dose of the bacteria, because most of the people are very much concerned about the dose of the probiotics, that how much they should take daily. So most of the countries, they have their own regulatory systems, and they said that the bacterial count should not be 10 to the power 8 CFU per day. And in our country, I will also tell you in the next slide that our country, we follow the FSSI. Then they also regulated that 10 to the power 8 cell is very much important to claim as a probiotics. What is the probiotic market in the all over the world? So I have just one report that in 2018 the probiotic market was $49.4 billion US, and they are expecting that in 2023 it will be $69.3 billion. And if you see the graph, that Asia Pacific particularly, which is in green color, you see that Asia Pacific region are occupying the highest probiotic market. So in our countries or in Asia Pacific countries, the probiotic market will be good in the future. Why it is so? Because you see that in 2018, Christian Hansen, they have expanded their production library for the development of next generation probiotic. DuPont in 2017, they have invested another $10 million US for probiotic facility in Madison, USA. And Kerry in 2017, another company, they have acquired the US-based probiotic player Gangadhar to enter into the probiotic market. So the giant player of the probiotic market in all over the world, they are expecting that there will be a big market in future, and they are investing a lot. If you think about India, we can say by 2025, India will be the 5th largest consumer market in the world. So India has a good potential to be placed in the probiotic market in future. What are the probiotic products are available in India? Probiotic curd, B active curd, Nesbita, probiotic ice cream, Yakult. So these all products are available in our country, and they are selling by all the dairy companies like Mother Dairy, Amul, Heritage Foods, Nestle, and Yakult. Even some pharmaceutical companies like Ranbaxy, Dr. Reddy's Laboratory, Zydus Cadila, GlaxoSmithKline, those they are also selling the probiotic in tablet or capsule or sachets form, and doctors are prescribing for taking this kind of probiotics daily for different for prevention of different diseases. And these are the pictures of all of you are known these pictures because you daily take these kind of products in your lunch or dinner. So in regulatory system of the FSSI, they said also that 10 to the power 8 cells to call as a probiotic is the mandatory. It should be more than that, not should be less than that. So 10 to the power 8 is the minimum number of cells requirement to call as a probiotic. Now coming back to the probiotic research in our department, last 3 decades we are working on probiotics and lactic acid bacteria in Anand Agriculture University at Dairy Microbiology department. Basically, we are working on indigenously isolated lactic acid bacteria and evaluating their probiotic potential, and we have developed acceptable product formulation and studying their stability and efficacy using in vitro and in vivo models. These are the pictures. It is our own products which are being sold here. So our product formats: we produce dahi, we produce lassi, we produce the buttermilk, we also produce raita-like products, and we also have the dried formulation as a capsules or as a powder form. These are our cultures which have been deposited in MTCC microbial type culture collection Chandigarh, and we have done the clinical trial for Lactobacillus helveticus, and we have also done the whole genome sequencing of these cultures. These are our product portfolio, what we have developed here: symbiotic dahi, symbiotic raita, symbiotic lassi, whey drink, herbal probiotic lassi, protein rich lassi, carbonated milk whey, acidophilus banana powder, acidophilus sweet malt powder, milk rice probiotic food, and we've also developed milk oat based probiotic lassi, and that technology has been transferred to the Swedish oat technology and Indian probiotic culture under the market testing by Hildu Functional Foods Private Limited. We have also done a clinical trial on our bacteria Lactobacillus helveticus MTCC 5463 with our very neighbor hospital, Karamsad hospitals, with the on geriatric populations. We have given this product to the geriatric volunteers, and we have estimated a lot of studies we have done, and we got very good results and promising results after feeding this product. We are also working on the bioactive peptides from fermented milk, camel milk, goat milk, cow milk, and soy milk, and these peptides are having different physiological benefits which are being published in different journals, and also we have submitted in the NCBI data bank. I just want to say that because we are talking about a lot about the antibiotic issues, antibiotic resistant pattern of the bacteria. Recently, within a year, we have started one trial on broilers, means the chickens. We have, in the poultry, basically they give the antibiotics as a growth promoters, but here we have not given any antibiotics. We have given the probiotics directly to the broilers, and we have compared the results. This T1 is the normal diet with antibiotics, but T2, T3, and T4, these are bacteria given in combined as well as individual, and we are getting at par results. So our bacteria is replacing the antibiotic, and we are getting antibiotic free meat and egg productions. These are the Lactobacillus fermentum and Lactobacillus plantarum. Another study we have done, the antibiotic associated diarrhea. Just I told you before that antibiotic associated diarrhea will cause the dysbiosis in your intestine. So here we have made a fermented rice beverage and given to the oyster rats, and our results, this is our product, A1, it is our product, and this is the normal control, this is the disease control and standard. You see, with normal control, our product is at par, but disease control, you see that there is a lot of aberration is there, or irregularity is there in the lamina propria and edema in the submucosa part of the GI tract. So it is the histopathology analysis of the colon. Another histopathology analysis of the same product we done on liver, and you see that our product is the fermented rice beverage is A1 and it is the normal control. If you compare with both, this is the at par, and compared to the disease control, you can say there is a lot of necrosis of the hepatic cells and periphery of hepatic lobules and congestion of the blood vessels. So we have a very good results by preventing the antibiotic associated diarrhea. Now I'm coming back to end of my presentation. This is the last slide. I want to say something about the future scope of the probiotics. Nowadays, the peoples are working mostly on the lactobacillus and bifidobacteria, but yeast also have a very good probiotic potentials, and we have made the fermented rice beverage by using Saccharomyces cerevisiae. Even Saccharomyces boulardii is another very good potent probiotics. So yeast could be another good ingredient, means probiotic bacteria for the probiotic organism for next or future probiotic, as a future probiotic. Liquid probiotic market will be higher than the any kind of sachets or tablets. So liquid probiotics are expected to dominate the global market, and replacement of the pharmaceutical agents provides a major opportunity for the probiotics. So the probiotics will help you to manage your health, so you should not take the medication for a very general cause. But high R&D cost for developing new indigenous probiotic is the need of the day, because most of the probiotics which we are using for different product formulations, they are importing from different countries, because I have listed that most of the countries which US FDA has given the approval, they are all outside our country. So we have to develop our indigenous probiotic strain. And scientific validation of the probiotic product is very important, as I have already listed the ICMR and DBT guidelines, what they have clearly made a very clear-cut chart that how you proceed to call your bacteria as a probiotic. And I have also showed you that Asia Pacific is projected to be the fastest growing region in the probiotic market. So we have a very good scope to work on probiotics and probiotic products. So lastly, I am in the Thanksgiving slides. So I will say that if you are happy today, you must give thanks to your gut, or gut bugs, means your probiotic bacteria, because they will help you to keep your immunity high and also to maintain your gut systems. Thank you all for listening me very patiently, and I am very much thankful to Mugberia College, because they invited me and to talk on the probiotics and how the probiotics is working to mediate the innate and adaptive immunity. Thank you all.
Thank you. Hello.
Hello.
Hi. Thank you, sir, for your nice presentation.
Yeah.
Also very much enriched and informative also. You have very much elaborately discussed with our seminar topics and themes also. Hope our students and our target audience is enriched enough with your informative talk. Thank you, sir, for sparing your valuable times with us. Thank you, sir. I hope so many questions has been written on the YouTube chat that will be discussed on question answer session. Now I would like to hand over this platform for conducting the second technical session to Dr. Apurba Giri, convener of this webinar. Apurba.
Yes.
Hello,
Ah,
are you getting my voice?
yes, yes, audible.
Dear participants, we are now going to start technical session 2. In this session, we have 3 speakers. At first, I will invite Dr. Maria Linear Silva. Dr. Maria Linear Silva is an assistant professor at Igas Moniz University, Lisbon, Portugal, since 2008 in nutrition and health sciences. Her bachelor degree is in nutrition science and master degree is in food safety and public health. She holds PhD in nutrition and metabolism at University of Central Lancashire. She lectures in metabolic biochemistry, food education, pathology, nutrition, investigation, methodology programs. Our main research interests are clinical nutrition and food compounds properties. She is master or PhD supervisor. She also do clinical practice in clinical nutrition since 2009. With this, I invite Dr. Maria for her valuable lecture on the topic food anti-inflammatory properties and its immune benefits. Madam, please start your lecture.
Okay, thank you. Have you seen my presentation?
Yes, please
Okay.
continue.
So good afternoon in India and good morning in Portugal. I would like to thank to Dr. Apurba and all of organization of Department of Food Processing in collaboration with Department of Nutrition and Research Cell for the invitation to participate in this webinar. I would like also to congratulate the thematic choice because this is a very actual topic nowadays. This is also my first presentation since March to begin the pandemic situation, what makes this international webinar a very special moment. So thank you for this also. This presentation intended to describe and summarize the role of anti-inflammatory properties in immune system. As we know, because there is very described before in others' presentation, coronavirus disease is a clinical syndrome caused by a novel coronavirus that leads to a severe acute respiratory syndrome, which emerged in China in December and then spread rapidly worldwide. So on March, World Health Organization declared a pandemic. The physiopathology of this virus is yet unknown. However, it is described that SARS-CoV-2 gains entry into the cells by angiotensin-converting enzyme 2 receptor, infecting predominantly the lower respiratory tract, binding to angiotensin-converting enzyme 2 on alveolar epithelial cells. So upon binding, there is a subsequent response of the immune system via an inflammatory-related manifestation and recruitment of antigen-presenting cells. COVID infection can manifest as an asymptomatic infection, or patient can present with the upper respiratory tract illness that may include fever, fatigue, and shortness of breath. So in severe cases, the most common complications are sepsis, heart failure, septic shock, and a disease is a very bad complication. Although the physiopathologic mechanisms are still not understood, it has been observed that the most severe and fatal cases with COVID have currently in elderly or in patients with underlying comorbidity, such as type 2 diabetes mellitus, cardiovascular disease, and chronic lung and others. These figures represent alveolar inflammation in COVID, in which figure A represents a mildew disease, which the adaptive immune response leads to a clearance of severe acute respiratory syndrome, coronavirus 2, from the lung. And the figure B represents a severe coronavirus disease, an excessive or uncontrolled innate immune response that leads a cytokine storm. And cytokine storm has been described as the most important characteristic of this virus regarding to the mechanism of action. So after COVID entry, host factors trigger an immune response against the virus, which, if uncontrolled, may result in a pulmonary tissue damage with functional impairment and reduced lung capacity, and damage to the pulmonary interstitial arterial walls indicates that an inflammatory response plays an important role throughout the course of this disease. During the COVID infection, the virus infects the antigen-presenting cells, macrophage and dendritic cells, which present SARS-CoV-2 antigen to T cells, leading to T cells activation, differentiation, and subsequent enormous cytokine storm release, as I said before. Furthermore, apart from respiratory failure, other common factors amongst critical COVID include the infiltration of monocytes, the decrease of lymphocytes, and a high level of inflammatory response due to pro-inflammatory cytokines, as I saw in the other presentation. In this context, it is apparent that viral infection-related inflammation and subsequent cytokine storm in severe cases plays a crucial role in patient outcome. So in healthy immune response, infected cells rapidly cleared, virus inactivated by neutralizing antibodies, and minimal inflammation and lung damage. But when dysfunction immune response, there are excessive infiltration of monocytes, macrophages, and T cell, systemic cytokine storm, a pulmonary edema and pneumonia, and widespread inflammation and multi-organs damage. Pathogens are recognized by cells of the innate immune system, such as macrophages and monocytes. So during COVID infection, the virus infects the cells, leading to cell activation and a subsequent cytokine release. The TLR receptor is a tool-like receptor recognized virus, allowing to enter in the dendritic cell, activation the transcriptor factors like nuclear kappa light chain enhancer of activation of the cell, and producing a classic inflammatory cytokine like tumor necrosis factors and interleukin-1 and others interleukins. That lead to a T cell activation which produce interleukin-2, which promotes a cytokine T lymphocyte activate. In this production, a differentiation of B lymphocytes to plasma cells, which produce antiviral antibodies. And these antibodies can bind to free virus, neutralizing them. What is important in this case? So the onset of progression of inflammation manifestation depends from balanced immune system, which is characterized by homeostasis state, and unbalanced immune system, which leads to a disease state. This depends on presence of risk factor that can be modifiable or modifiable, and target drug treatment and or a healthy lifestyle in which dietary patterns plays an important role. In this context, all potential therapies Prevention strategy that may reduce incidence and severity of infection are of vital importance. Thus, some of anti-inflammatory therapies currently are promising in combat of COVID. Likewise, the potential role of nutrients and foods that may exert an inflammatory and immunomodulatory effect has been explored. So the question is what is the potential role of food compounds against COVID? Many studies, very recent studies, has been reported, and the meta-analysis reveal that there are evidence that properties of some food compounds may hold some promise of respiratory infection improvement in COVID, such as vitamin A supplementation improve clinical symptoms of pneumonia. Vitamin C supplementation also decrease the incidence of pneumonia. And vitamin D supplementation protect against respiratory tract infections. All of these cases in COVID infection. The zinc supplementation shortened the duration of symptoms, and the deficit of some minerals such as copper, zinc, and iron contribute to the susceptibility of current respiratory tract infection. The vitamin A has been reported as modulates T cell function throughout directly impacts T cell membrane integrity to improve signal transduction and cell division, and also indirectly by affecting inflammatory mediators generated from other immune cells. But also the omega-3 and omega-6 fatty acids are capable to partly inhibition many aspects of inflammation, including leukocytes, chemotaxis, adson, molecular expression, and these fatty acids inhibit the activation of pro-inflammatory transcriptor factor nuclei and reducing the expression of inflammatory genes, and activation the anti-inflammatory transcription factor. So this is very important. But finally, the phytochemical compounds that are present in natural foods, which can be successful, used in order to prevent, control, or block disease progression by target inflammatory mediation. Thus, all of these nutrients or compounds of food with a high anti-inflammatory capacity are expected to contribute to a better immune system. On the other hand, it is very important to note that the low micronutrient status, such as vitamin A or zinc, has been associated with increase of infection risk. And frequently, poor nutrient status is associated with inflammatory and oxidative stress, which in turn can impact the immune system. Moreover, it is very important to note that the excess of amount of these nutrients also impair immune function. So the doses is very, very important in this case. As I mentioned before, in the first slide, the inflammation associated with pre-existing comorbidity was highlighted and a significant risk factor for COVID patients. So different studies have demonstrated that COVID patients without other comorbidity, but with diabetes, were at a higher risk of severe pneumonia and an excessive uncontrolled inflammation response. Furthermore, serum level of inflammation related biomarkers such as interleukin 6 and C reactive protein with were significantly higher in diabetic patient, comparing with those without, suggesting that patients with diabetes are more susceptible to an inflammatory state. In addition, there is evidence of increased incidence and severity of COVID in patients with diabetes. So COVID could have effects on the pathophysiology of diabetes. In this context, I suggest that blood glucose control is very, very important, not only for patients who are infected with COVID, but also for those without this disease. In biochemistry laboratory of Egas Muniz, it has been developed on different studies in which have demonstrated the effect of different plants or spices in blood glucose level. So the first study, one of those is cinnamon, a spice that have demonstrated in different studies a significantly decrease of postprandial blood glucose level, especially significantly reduce the maximum concentration after an oral glucose tolerance test in non-diabetic adults. There are very mechanism of action that has been proposed to this effect. However, one of the mechanisms that this group proposed, that is the effect of cinnamon on glycemia, may be related to the insulin action throughout increase of insulin receptor. However, it is very interesting to note that another study with cinnamon, extract of cinnamon, revealed that in type 2 diabetic adults, the results are not conclusive because there is no interaction between the independent factor and repeated measure factors. So it means that is not correct comparison group. So this is mean that cinnamon infusion did not demonstrate a significantly effect on area under the curve and maximum concentration and variation of maximum concentration during oral glucose tolerance test after ingesting the cinnamon infusion. So it means that in diabetic patients, maybe cinnamon have not the benefic effect, has non-diabetic effect. Regarding to cinnamon powder, we also have different studies, and the results of our lab revealed that also, as I said before, did not significantly decrease postprandial glucose when added to a dessert, in this case in the custard tart, in healthy adults. And another study, it was verified that when added to a semi-solid high sugar meal, in this case 2 meals, did not significantly affect the postprandial blood glucose level on healthy subjects also. So it seems that probably cinnamon Burmani powder demonstrated not significantly affecting short term, because these results are made in oral glucose tolerance tests once and not in a long term. So maybe no significant effect on short term on glucose homeostasis, especially here in non-diabetic adults. Ginger also demonstrated its potential use in glycemia. So ginger infusion significantly reduced blood glucose area under the curve and also the maximum concentration during the oral glucose tolerance test and the variation of maximum concentration. So the intervention group that ingested ginger demonstrate that significantly reduce these parameters when compared to control group in non-diabetic adults. These data are not published. It's a clinical nutrition thesis. The baobab has cinnamon or ginger also show that can reported a benefit effect on glycemia control because significantly reduced postprandial glycemia area under the curve. So, given the laboratory results of this study regarding to its compounds, it is proposed that hypoglycemia effect of this fruit is mostly due to its procyanidins content, a very important compound present in fruits. So the results of both clinical and laboratory data reveal a potential use of this fruit has an element of pharmacological interest for the prevention and or treatment of type 2 diabetes mellitus. Furthermore, taking in account that blood glucose control is very important, not only for patients who are infected for COVID, but also for without the disease, the ingestion of these plants and spices. It is recommended to protect against COVID. Of course, in the correct doses. In terms of conclusion, at this moment, we know, and this is very important to know, that no effective treatment for COVID at the moment. The inflammation process has been associated with pre-existing comorbidity, representing a very important risk factor for COVID. Furthermore, nutrients and compounds with anti-inflammatory properties may prevent or attenuate the inflammatory and vascular manifestation associated with COVID, and a healthy and balanced diet can offer a necessary macro and micronutrients and can restore and maintain the immune cell function, and this is very important. I would like to say that supplementation above the recommended dietary allows, but within recommended upper safety limits for specific nutrients is also very important. Finally, it must be noted that to date there are no known evidence-based therapeutics or treatment strategies. Likewise, there is no single food or natural that has been demonstration to prevent COVID, which has been made clear by World Health Organization. So more studies is very important to make to determine that. I would like to acknowledge to Egas Muniz for financial support, research, and I would like to thank to Biochemistry Laboratory to operation support, and thank you very much for our attention.
Thank you. Thank you, Dr. Maria, for your enriched and informative presentation. I think our participants have enriched enough regarding the food anti-inflammatory properties to get immune benefits. Now, I will invite our second speaker of this technical session, Dr. Alexandra Bernardo. Dr. Alexandra is a senior associate professor at Egas Muniz University, Lisbon, Portugal. She holds PhD in physical chemistry. She has more than 20 years of teaching experience, both in BSc and MSc health-related degrees. She is the vice coordinator of the Clinical Nutrition Master degree since 2007. Her main research interests are clinical nutrition, environmental health, and more recently, forensic document analysis. With this, I invite Dr. Alexandra for her valuable lecture on the topic Food Antioxidant Activity, a Potential Property for Immune System. Dr. Alexandra, please start your lecture. Hello, Dr. Alexandra.
Okay, thank you very much for your kind words. I would like to confirm that you are all seeing my slide, please.
Yes, please continue.
Okay, so good afternoon or good morning, depending from where you are attending this webinar. I am Alexander Bernardo, as mentioned before. I am a professor and researcher at Egas Muniz Higher Education School. And first of all, I would like, in my behalf and in the name of Egas Muniz teams, to thank the Department of Food Processing and the Department of Nutrition and Research Shell of Mugberia University, namely Dr. Sampan Misra, Dr. Purba and Dr. Bitan Samantha, for kindly inviting us to take part in this webinar, which gave us the opportunity to share our work, but above all, to learn with other distinguished speakers about this very exciting and actual topic. As we all know, what brings us here is the actual global concern about pandemic coronavirus infection, which rapidly spread across the globe. In the early beginning of this highly contagious respiratory infection, many researchers and medical doctors turned their attention in order to better understand the body's impact and the various interactions that stopped entire countries and continents. The first studies that try to characterize metabolically and physiologically the impact of this virus reported that severe respiratory distress is associated with increased cytokine levels and lymphopenia, as mentioned in the previous lecture. Cytokines and chemokines have an important role in immunity and immunopathology, and exuberant immune responses have been shown to potentially cause lung damage and diminish survival. As illustrated by Patterson and Ho in the early pandemic stage, as represented here, two scenarios were described when compared to an infected individual that you see in the left panel. One, a moderate scenario that exhibits an increase in EL6 and a decrease in CD4 and 8 T cells that you see in the middle panel, and another one where a further increased production of EL6, EL2R, EL10, 10 alpha, while T lymphocytes decrease in the right panel. This pro-oxidative and pro-inflammatory state is referred by the authors, as mentioned here before, as a cytokine storm, and is associated with pulmonary damage, respiratory distress, and adverse outcomes. So far, the screening carried out in the different countries confirmed this pattern, where individual response to COVID-19 varies widely. Some people have no symptoms at all, while others become so sick that they eventually need mechanical assistance to breathe. The clinical data also revealed that the risk of developing dangerous symptoms because of COVID-19 may be increased in old people and also in people of any age, but that have other health problems like the heart, lung conditions, weakened immune system, severe obesity, or diabetes. Having in mind the mechanisms described in the previous presentations, by my colleague Leonor and the other speakers, several authors propose nutritional approach in the COVID prevention, given that energy, substrates, and regulatory molecules from diet may modulate and improve the immune system, as sustained in the Calder work published in Nutrition, Prevention, and Health, and also by the work of Derbyshire paper. Regarding the nutrition approach, once again, this coronavirus presents us with a multi-level challenge where a multi-level framework for action was proposed by Naja and Ahmed. As we all know, the influence of this pandemic has already gone behind the individual and the community to reach national and global levels. A particular feature of this pandemic is highlighting the interdependence of these several levels, whereby the health of the individual becomes a direct function of his own awareness and choices, the units of the community, the preparedness of the government, and the global engagement to face this threat. In this perspective, a framework for action and recommendations are needed for each of those levels. In this context, the authors proposed recommendations on nutrition and food security at the individual, community, national, and global levels to mitigate the impact of COVID-19. For the moment, the recommendations consist in maintaining the physical and mental health of individuals, resilience of communities, and national and global food security. Specifically at the individual level, well-balanced meals where food rich in vitamins A, C, E, B12, zinc, and iron should be present, and the common denominator that drives most of nutrition and dietary recommendations to combat viral infections, including this one, COVID-19, lies within the link between diet and inflammation and diet and immunity modulation. Besides nutrition concerns, it is also recommended health lifestyles like exercise, regular sleep, and meditation. Regarding the literature information, oxidative stress and chronic low-grade inflammation in the lungs are associated with aging and may contribute to the age-related immune dysfunction and mortality risk. On the other side, existent evidence highlights that diet has a profound effect on people's immune system and disease predisposition. It has been suggested that specific nutrients or nutrient combinations may modulate the immune system response, as already described in the previous presentation. In this context, Portuliano suggests that along with vitamins and minerals, other natural antioxidants contained in foods such as polyphenols can play an important role in this pro-oxidant and pro-inflammatory state, where these specific bioactive compounds can target oxidative stress and inflammation. In fact, polyphenols are phytochemicals which are found in food and are separated from essential micronutrients because a deficient state has not been identified. However, these chemicals play an important immunomodulatory role. As mentioned in 2011 Portuliano work, these dietary antioxidant molecules can be found in olive oil, such as alpha tocopherol and several flavonoids as tyrosol, among others. In turmeric, which has an important antioxidant properties due especially to its content in curcuminoids, and in red grapes and wine, where resveratrol is associated with relevant immunomodulatory activity. The growing interest in polyphenols and food antioxidants properties is visible in the graphical representation in 2017 by persons, where a significant increase in the number of papers per year was found on the Web Science database using the search item food and antioxidants. Most of these papers explored total antioxidant activity, individual antioxidant molecules identification, as well as several antioxidant assays such as DPPH, ABPS, ORAC, FRAC, and TEAC. Polyphenols are indeed strong antioxidants that may intercept single oxygen, prevent first chain initiation by scavenging initial radicals, binding metal ion catalyst, decompose primary products of oxidation to non-radical species, and break chain reaction in order to prevent continued hydrogen abstraction from substrates. In 2015, in an exhaustive review, several phenolics and polyphenolics in food, beverage, and spices were systematized and health effects reported. These compound structures range from simple phenolic molecules to high complex molecular weight polymers like procyanidins. Sorry. This is okay. Okay. Authors classify natural phenolic antioxidants in major 6 groups: phenolic acids, flavonoids, stilbenes, coumarins, lignans, and tannins, where flavonoids, stilbenes, and tannins are associated with relevant roles in health antioxidant activity. It is also reported that polyphenols can actually function as co-antioxidants and are involved in the regeneration of essential vitamins. In addition, other mechanisms such as inhibition of xanthine oxidase and elevation of endogenous antioxidants were also described. In the same year, 2015, an interesting review related functional components and medical properties of food. In this review, authors identify functional ingredients, their sources, and potential benefits such as anthocyanidins, catechins, flavonoids, lignans, and tannins, and recognize carotenoids, polyphenols, phytosterols, and tocopherols and tocotrienols as antioxidants, anti-cancers, and immune-modulating agents, essentially due to their major ability to neutralize free radicals and reactive oxygen species. Concerning the immune modulation mechanism, authors propose the multiple antioxidant and active oxygen neutralize capability, which allows these compounds to reduce the deleterious effects of reaction oxygen species and free radicals, which cause premature death of human cells. Two pre-COVID papers from last year provide actual overviews of the food antioxidants' relevance in health and in particular in modulation of immune function. Durazo overview presents an update of each class of phenolic compounds and their properties, as well as a picture of key concepts in polyphenol research, and they highlight new frontiers for polyphenols in nutrition and health. The use of existing databases of bioactive compounds with health relevance, including polyphenols, is also described as a key tool for human health research. As future investigation, the authors propose that polyphenols research should be addressed towards two main directions: the identification of new structures of polyphenols through innovative advanced technologies, and the studies of bioactivity starting from evaluation of interactions between in vitro assays throughout the studies on cell lines and animal models to bioavailability and intervention studies until epidemiological and clinical trials in humans. The use of nanotechnologies is also proposed for micro and nano delivery polyphenol target formulations, enhancing the polyphenol's therapeutic application. On the other 2019 paper, the immunological effect, mechanisms, and clinical relevance of several vitamins and oligoelements is explored, as well as a green tea relevant flavonoid, the epigallocalactin 3 galate, that have been shown to be effective in maintaining or improving immune function, including inhibition of pro-inflammatory mediators, promotion of anti-inflammatory function, modulation of cell-mediated immunity, and communication between the innate and adaptive immune systems. However, the authors highlight that studies are not always consistent with preclinical animal models, and the immunomodulatory effects have not yet been examined in humans. This is mainly due to the great variation among human studies, the doses used, and the populations of study, demonstrating a need for more uniform clinical trial designs, better characterized populations, more information about chosen dose, and more consistency in the outcome measurements chosen. The described antioxidant properties have been studied by our research group, along with master and PhD students, where the total phenol content, the TPC, as well as antioxidant activity in several commercial and manufactured foods were characterized, and their ingestion effects in health were also explored, as my colleague Leonor Silva described in her presentation. Here I briefly share some of our published results, and in the present work here, Commercial and manufactured equivalent juice were compared, and we found that the ascorbic acid added to commercial juice formulation increased significantly both TPC and antioxidant activity. In another study, we investigate the antioxidant content and properties over time. The results obtained suggest that immediately after manufacturing execution or opening of the package, the juices present significant difference in the content of total phenols content, TPC, and FRAP values, with higher values for the commercial juice. During storage until 72 hours kept at 4 degrees Celsius, we observe that the antioxidant properties are almost constant. We also investigate solid foods such as Barbella wet, where the tocopherol content is higher than the same wet species from different regions. We also observe that the antioxidant activity increases with the mechanical processing of grain, probably happens due to the greater exposure of the chemical constituents. In a different study, we found that Physalis peruvianus flavonoids represents more than half of the phenols content in the fruit extract. In this study, we have also characterized antioxidant activity through FRAP and ABTS methods. We also characterize other fruits as baobab, which was brought by an Angolan student from our country, and the commercial tomato juice. In these studies, we have characterized the proanthocyanidins and tannins content beside the TPC in the case of the baobab, and both lipophilic and hydrophilic fractions of tomato juice. However, our major core interest is cinnamon. We have characterized the TPC and the total antioxidant status of a typical custard tart, Pastel de Belém, very appreciated in Portugal, and compared with custard tart plus 3 grams of cinnamon burmani. We found that after cinnamon powder addition, the TPC has increased almost 6 times and the antioxidant activity 2 times. We also characterized the TPC and the TAS from cinnamon infusion, and we found a significant amount of the total phenol contents and antioxidant activity. At the moment, we are studying the antioxidant properties of cinnamon, bitter melon, ginger, and baobab extracts, and these effects in cellular proliferation of 2 cell lines of colon cancer. This is the master thesis of work of Cláudia Duarte, which is supervised by me and my Egas Muniz colleague Alexandra Maia Silva, in collaboration with Cristina Albuquerque and Lucília Pereira from Lisbon Oncological Hospital. I hope that I have guided you on a journey that begins with a global threat to individual and social health and led us to one of the possible weapons to protect ourselves and eventually fight it against COVID-19 and other infections concerning the immune and the anti-inflammatory targets. Let's hope that this adventure will be short and that soon the distance between us will be shortened enough so we can continue this journey together and especially close to those we love. In the last slide, you can see again Egas Muniz campus, where you are all welcome. I want to acknowledge Egas Muniz for all the financial and operational support to our research. I also want to thank all the group members and students, and especially Professora Fernanda Mesquita for sharing with me during 20 years knowledge and friendship, as well as the opportunity to work in the area of clinical nutrition. And finally, thanks very much for your attention. Okay. Yes.
Hello. 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 Are
বলুন।
you getting my voice?
হুম,
Uh,
বলুন।
Thank you, madam, for your valuable presentation. I think antioxidant activity of food is very important property for immune system. I hope our participants are engaged with your lecture. Now I will invite our third speaker and the last speaker of this technical session, Ms. Koel Pal Choudhury. Ms. Koel Pal Choudhury is research dietitian in IPGMER and SSKM Hospital, which is known as PG Hospital in Kolkata. She is MSc in Food and Nutrition from Calcutta University, Master in Social Work from IGNU, Certified Diabetic Educator from Dr. Mohan Diabetic Institute, Chennai. She is also guest lecturer in Haldia Institute of Health Science, IGNU, and Bangladesh Academy of Nutrition and Dietetics. She is consultant and community nutritionist in Ramkrishna Medical Hall Nursing Home, Ritik Nutrition Home, Ramkrishna Mission, Bharaka Public Trust NGO. She received IDA Award, NSI Award, West Bengal State Science and Technology Congress Outstanding Paper Award. With this, I invite Ms. Koel for her valuable lecture on the topic, the importance of immune maintenance balanced diet in pandemic and post-pandemic situation. Madam, please start your lecture.
Thank you so much. I'm audible.
Yes, continue please.
Thank you. Yeah. Good evening and namaskar to all. And thank you for the excellent program, excellent and enriching webinar. So I am starting my topic. I am Koel Pal Choudhury, dietitian from India. Nowadays, we are very cautious about COVID-19 diet. Yes, it is very trending nowadays because people now consider prevention is better than cure. Now we are thinking about the phenomenon of importance of food science. Rather, we are thinking about the implication of dietetics in our daily life. Whatever.
Excuse me.
Now we
excuse
are.
me.
Yes.
Excuse
Yes.
me, man. Please on your video. Please.
Actually, I am from phone, so that's why the video is minimized.
Okay. Continue.
Okay, thank you. So nowadays we are used to hear one word, that is immune boosting foods, right? But what is immune boosting food? Rather than, I want to say one thing, that if we have the marginal immunity power, so that's why how can we go for immunity boosting food? We have to focus first on immune maintenance food, and afterwards we can focus on immunity boosting food. Why so? Because I am describing today what is immune maintenance, and then why should we focus on immune boosting food? The answer lies here. The immune maintenance diet means the balanced diet, because we have to enhance our first line mechanism, means we have to empower our immunity. And how is it possible? From balanced diet it possible. In the picture you can see that there are 50% fruits and vegetables, 25% cereals and 25% protein. I will let you know at the end of my lecture why I am giving this picture. Nowadays, we are hearing about many fancy diets like keto diet, intermittent diet, alkaline diet, and so on in Indian perspective. It's there is a lots of clinical trials we know, but it's not suitable for everybody. Just remember it, because we have to focus on the balanced diet because it is suitable for everyone. First of all, definitely it's variable from person to person, but the main thing, the main kind of balanced diet is it consists of the macronutrient, means carbohydrate, protein, fat, and the micronutrients, mean vitamin, minerals. And along with that, fiber, probiotics, and other things are also there. Water is also there. Hydration is very important nowadays. But today, my focus point is I'm trying to brief the significant connection between nutrients and immunity. What's that? I want to brief you by some scientific evidence. So let's start. Carbohydrate, one of the major macronutrient. What is the relationship between carbohydrate and immunity? I am explaining now. By class 2 MHC, it is present in T cell, and carbohydrate has the power to recognize the foreign and self antigen also, because it has the nature of adaptive immunity too. Yes, it is existent in different kind of like prokaryotic, eukaryotic cell. So carbohydrate has the power to adapt the adaptive immunity. Switching to my next slide, here is also seen that if we focus on the glycobiology, then it is seen that the glycoprotein mechanism means glycogen plus protein combination. It's a very complex pathway, but it is related to innate immunity. It has been highlighted that, in short, carbohydrate and protein interaction help in pathogen invasion and definitely host immunity. So definitely I can conclude that from some scientific evidence that carbohydrate is included in innate immunity as well as the adaptive immunity. Now, if we focus into the protein part, obviously the main macronutrient in order to immune system, the crown goes to protein, definitely, because the protein is consisting of different kind of amino acid. In short, I can tell you that the protein energy malnutrition, it's a very familiar term. And nowadays, the latest term is protein energy wasting, is PEW, protein energy wasting, which actually impairs the immunity, particularly T cell system. And obviously, it increases the infection as well the inflammation. So obviously, the exact high biological value protein is needed in our body. And coming to the amino acid part, the key factor of amino acid is it helps in our growth and development. So amino acid has its big role in case of immunity too. And from BCCA, means branch chain amino acid, to whey protein, they have some particular activity in immunity. In next slide, I am going to focus some of the amino acid which has immunity boosting effect, like alanine. Alanine helps in prevention of cell death. Arginine accelerates WBC, means white blood cell functioning. Glutamine eliminates the pathogen. Glutathione, it shows antioxidant and anti-inflammatory effect. And isoleucine and valine, these are the two branch chain amino acid which acts in immunity system and reduces inflammation. And another is conditioning amino acid, means taurine. Taurine actually found in leukocyte, and it precedes its activity also. And last but not the least is threonine. Actually, it produces the T lymphocytes. So from the protein part, we also seen that the amino acid play a very important role in immunity system. So carbohydrate, 50 to 60% of carbohydrate from daily diet, from regular calorie is needed, and as well as the high biological value of protein. Protein is actually dependent upon the body structure and the weight of an individual. So carbohydrate and protein is equally important in our daily diet. Now I'm switching to the lipid. People are getting confused about the fat part. People are reluctant to take fat. But remember, the minimum fat is needed for our body. Why so? In terms of immunity, we can say that essential fatty acid is required for growth and development, and the free fatty acid part help to active the immune cell. So essential fatty acid is equally important for our body, and as well many vitamins. They are fat soluble vitamins. That means A, D, E, K. These are the fat soluble vitamins. So fat is needed. If I'm switching to my next slide, we can see that fat is stored basically in adipose tissue, right? So which secretes the cytokine like hormone, that is adipokines. We all know the patients who were affected in COVID-19 or coronavirus, the CKK means cytokine storm are very evident. And like what adipokines is affected our patient, and adipokines main role is to bridge the metabolism and the immune system. So adipokines has its own kind of work. And as for example, I can say you leptin. If we see the leptin mechanism, leptin pathway, we can see that it helps in energy metabolism. And in brief, it helps in immunometabolism, both in innate and adaptive immunity. So we can conclude that in our daily diet, we need the carbohydrate, we need the protein, we need the fat. Along with their own functions, they play a very important and highlighting role in terms of immunity boosting. So in case of balanced diet, we have to focus on the main macronutrient. And definitely alongside, we have to focus on the vitamin and minerals part. So in case of vitamin and minerals, we can focus on the vitamin A, C, E and zinc, which work as epithelial barrier, which work in the epithelial cells. And both fat soluble vitamins mean A, D, E, K, B complex, iron, zinc, copper, selenium, all help in antibody production. All the vitamin, minerals, all the macronutrients help in antibody production and boost our cellular immunity. So along with micronutrients is also important. Macronutrient is for maintaining our daily diet, and micronutrient is for boosting our immunity. So we need both for maintaining and boosting our immunity. So now I am going to the second part of my presentation. We have to focus on the balanced diet. Yes, there is lots of diet, what I said, but it's not suitable for everybody. What I said in earlier, that if we consider a round plate, then 50% should come from fruits and vegetables, that is vitamins, minerals, fiber. 25% cereals means carbohydrate, and 25% from protein. Likewise, milk products, and meat, fish, egg, these are the things. If we consider this in our daily plate, then it will be a good balanced diet for an individual person. Now, apart from immunity maintenance, we are focusing on the immune system booster. Yes, there are lots of immune booster things like zinc, iron, vitamin A, vitamin E, vitamin C, and vitamin B6. Their functions as well, they act as antioxidant, anti-inflammatory effects. They have anti-carcinogenic effects too. So these are all important in our daily diet. As per Indian perspective and from a dietitian desk, I can include something to boost your immunity. Likewise, there should be some nuts like almonds, walnuts, sunflower, pumpkin seeds. They have all the bioactive compounds which help to boost your immunity. And as well some herbs like turmeric, ginger, garlic, clove. These help to boost and maintain your immunity system. And take some good beverages like herbal tea instead of black tea. Sometimes you can take lassi. So this can be included in your daily diet along with the good cereals, complex carbohydrate, high biological value of protein, and definitely a lump amount of vitamins and minerals, which comes from fruits and vegetables. And last but not the least, the main suggestion, you have to work on your exercise, because exercise helps to escalate your immunity power. And obviously, it helps to increase your metabolism. And as well, like the senior speakers talk, we have to reduce the intake of smoking, alcohol, this kind of thing, because it suppress our immunity, it damage our DNA and all the synthesis, all the mechanism of our body. And as well, we have to focus on the fiber for our good metabolism, good bowel movement, and obviously probiotics for our gut health to escalate our gut microbiota. So balanced diet, exercise, reducing stress, you have to go for exercise and as well as some yoga, some meditation for maintaining a good and healthy diet. So balanced diet is for your maintenance, and there are some immunity boosting material which can help your first line mechanism, which can help to heighten your first line mechanism. It can heighten your immunity. So balanced diet is important. So stay healthy and secure your immunity. It's very important in this situation and fight for the coronavirus. Thank you so much, all of you, and respected speakers, organizer, and audience for your kind attention. Thank you so much.
Thank you, madam, for your lecture. I hope we should more stress on this immune maintenance balanced diet. I think our participants are engaged with your lecture. So now the session is open for question and answering. Due to limited time, we'll go for some selected questions. We have 6 speakers, eminent 6 speakers. So many questions they have arised our participants. Only few questions I will ask. At first, I will ask our first speaker, Dr. Gomes. Momoni Bandyopadhyay, she asked, raw vegetables are good source of dietary fiber. Is that safe to eat without boiling or we have to boil? Sir, please, what do you think?
Unmute, please unmute.
Okay.
Yes.
You
Yes.
can hear?
Yes, please.
You can hear?
Yes.
Momuni, it's a good question that how we should take the raw vegetables. Actually, you bring the vegetable from the market, wash thoroughly and properly in cold water, and keep it in daylight for 24 to 48 hours as possible. Don't put it in the fridge. And then you can have some of them. Normally what we take, cucumber, tomato, all these you can take raw.
Sir, one more question. Bonasri Rai Chaudhary, she asked, can we take vitamin supplement from outside or from normal diet?
Yes, Manusri, that's a very good question. I'm sorry, I forgot to tell you that during my lecture, that during this time, crisis time, all of us should take an extra quota of vitamins and minerals. I can name just one, that Becadexamine is a Glaxo company. You can take one capsule after dinner, you take for 15 days, give a halt for 1 month, again you can take for 15 days. That will definitely help you, because from the food, we may not get everything what we expect. That's a good question. Thank you very much.
Now, I'll ask Dr. N.M.N.K. Narayana. What type of nutraceuticals or functional foods need to be developed in present era of COVID-19? That question is asked by Sudipta Jana from Vidyasagar University.
Yeah,
Madam, please.
Yeah, actually, as I said in my presentation also, since there is no cure for this disease up to now, we have to think about immune boosting foods. I think that is the way that we have to think. That means immune boosting foods should be developed, and then by increasing our immunity, we can avoid. Maybe we can have some help to prevent this infection of this disease.
Okay, madam. Uh, One more question
Yeah.
from Assam Agricultural University, Dr. Saida Nasar. He asked, will whatever the phytochemicals you told about, will these phytochemicals rich food help to recover COVID-19 patient?
Actually, the recovering of the food, actually we have to study it properly, because when we normally now we give western medicines also, but without any the known effect. So we have to study it before telling that whether we can recover from this COVID-19, because it doesn't have still better treatment. That is the problem, I think. So I think to recover, we have to study properly the scientific uh, facts, whether we can do it.
Okay. Same person asked one very good question to Dr. Hathi, that really this coronavirus, it is a microorganism, and your talk is a probiotic, that also a microorganism. So would probiotic boost our respiratory system that is more prone to COVID-19?
So means just I want to clear the questions again, that you
He's
want to,
asking, could probiotic boost our respiratory system
yeah,
that is
yeah,
more prone to COVID-19?
yeah. I have given one or two studies that already that study was conducted in China. They have given some office goers peoples to probiotics, and they said that the upper respiratory tract infection, UTI or RTI, respiratory tract infection, is being reduced. Even and a few studies have already done on children also. Those basically suffer from some flu. That probiotics are when they have given the probiotics along with the medications, that the chances of respiratory tract infections is reduced because of the immunity or immunomodulatory activity of the probiotics, because they produce IL-10, IL-1 beta, leukocytes, and other immunomodulatory cytokines. So your immunity will get boosted up, and these kind of viruses, they won't allow through entry through these epithelial cell lines. So probiotics will definitely will help. But if you say the COVID, that this is the another, I can't say right now, because there's no direct study on COVID, because the COVID has been just started a few months back, and there is no evidence or data that I can say yes, the probiotics should directly fight against the COVID. I can't say right now.
Dr. Hathi, one more question.
Yeah.
Dr. Kalyani Saha from ICMR, she's asking, is there any difference in effectiveness of homemade or commercial packaged card? I know you are a dairy technologist background, so I think you will be you will able to say that.
So it's a very good question, and most of the people usually ask these questions, that the curd or dahi which we are preparing in our household level, really it have some benefits, or we should consume the commercially available like Mother Dairy or Nestle or other dairy plants. They are selling different. If you say about the probiotic particularly, I will say in domestic or at a household level, it is not possible to produce the probiotic. But for curd, again, I am telling you, because I am right now I am in Gujarat, in the western part of our country. So here we have one project sponsored by the ICAR. Basically, the project is to see the bacterial counts in the domestic dahi prepared in the household levels, and we have analyzed the bacterial or microorganism profiles of those dahi we have collected from different places of Gujarat, and we have found that 90% of dahi are produced with the enterococcus, not the lactic acid bacteria. It's a very serious now, because most of the dahi are producing at a household level, they are predominant with the enterococcus. I'm not telling that enterococcus is going to harm you, but thing is that enterococcus doesn't have the GRAS status. GRAS means they are not generally recognized as safe, and maybe the enterococcus contains some pathogenic islands, means the pathogenicity. So compared to the enterococcus, lactic acid bacteria are more beneficial, and they have a GRAS status, and since long people are consuming lactobacillus. So I will suggest, if you can afford the dahi from the commercially available in your nearby places, you must go for buy it, because ultimately your health is very important for you. So compared to the domestic, because you see the domestic level, what are you are producing, there is no defined strain, because you can't say that how you are preparing oil. But in the commercial level, you know that these probiotic bacteria are coming, or these lactic acid bacteria are going in your systems. So definitely I will suggest you go for the commercially available product.
Okay, one more question, Dr. Hathi. It is last question for you. Ashok Senapati is asking, is there any adverse effect of probiotic?
Yeah, it is. Yes, it is there. Some I have across some literature, I have come across a literature. Those who are having the immunocompromised conditions, the persons who are suffering from immunodeficiency, and their gut flora is not properly restored. So if you are giving the probiotic bacteria, the body may react as antigens. So for this, but most of the cases, probiotics still did the lactic acid bacteria and the bifidobacteria, which I have delivered my talk recently, that is totally safe. There will be no any kind of negative effect of the probiotics. But those who are basically the persons who are suffering from immunocompromised conditions, their body may react with the probiotics and may have some problems.
Okay, I think okay, I think there is not too much question, and also we don't have also Sir Dr. Anthony Gomes, you want to say something?
My last answer to Dr. Swadesh Ranjan, he asked me which sea fish may help us.
Yes, please.
To tell you that mackerel, sardine, these are the fish available in the Medinipur district, from Chandipur, from Digha, and from Orissa. So these are coming to the market, but the Bengali people, they are not accustomed taking this fish. So you should start eating, apart from Rui Katla, you should start taking this sea fish. Okay, thank you.
So I think we have a very limited time. Already too much time we have taken from the participants.
Video is stuck.
বিধান বাবু,
হ্যালো,
আপনি কন্টিনিউ করুন। অপূর্ব বোধহয় ইন্টারনেট কানেকশনে আসছে না।
হ্যালো,
হ্যাঁ, ডক্টর বিধান সামন্ত, ইউ কন্টিনিউ, ক্লোজ দিস সেরেমনি।
হ্যাঁ, hello, hello. So the question answer session is now closed here. Audience participants are requested to write their questions in our WhatsApp group, and we will communicate to our speakers. Now the time is coming. Vote of thanks. Our
Hello. Hello. Yes.
Apurvo,
Yes,
can you hear?
Yes.
Can you hear me?
Yes, I am
Hear
getting.
me?
Hello. I am getting. Hello.
Okay. Any more question, Apurvo? Any more questions?
No. There we have very limited time, so we are not going to for further question answering. So I am requesting Mona Lisa, please go for vote of thanks.
What?
Sir, am I audible?
Yes, yes.
Yes, audible.
Good evening, Honorable Chairperson, respected convener and joint convener, our most valued invited speakers and dear participants. It's my privilege to have been asked to propose a vote of thanks on this occasion. First of all, I would like to propose a hearty vote of thanks to the Chairperson, Dr. Shapon Kumar Mishra, Principal of Mugberia Gangadhar Mahavidyalaya, for gracing today's international webinar and delivering the inaugural address. I, on behalf of organizing committee members, would like to thank all the speakers, Professor Anthony Gomes, Dr. N.M.N.K. Narayana, Dr. Subroto Hathi, Dr. Maria Leonor Silva, Dr. Alexander Bernardo, and Ms. Koilpal Choudhury to spend their valuable time from their busy schedule. I hope that participants must have benefited with their excellent presentation. I would also like to thank the convener of this webinar, Dr. Apurva Giri, Coordinator, Department of Food Processing, Assistant Professor and Head, Department of Nutrition, and Joint Convener, Dr. Bidhan Chandra Samanto, Coordinator of Research Cell, Associate Professor and Head, Department of Chemistry of Mugberia Gangadhar Mahavidyalaya, for their contribution and guidance. I wish to extend my thanks to all the advisory committee members of this webinar, especially to Dr. Mesquita from IGAS Moniz University, Portugal, Dr. A.K. Chauhan from BHU, Dr. S.T. Kanjiala from NDRI, Dr. B.V. Venkatasya from Dairy Science College, Bangalore. I also offer my sincere thanks to Dr. Kalipoto Maiti, Coordinator of IQAC, all the teaching staff of Mugberia Gangadhar Mahavidyalaya, including all the faculty members of Department of Food Processing and Department of Nutrition. I would like to thank especially Mr. Khokon Chandra Gayan, ex-student of Nutrition Department of this college, for his continuous hard work for organizing this webinar. I would like to thank our participants from India and abroad, especially from Sri Lanka and Portugal, for their active presence, support, encouragement, and constant cooperation. Thank you all.
Thank you, Mona Lisa. And again, thanks to all participants, speakers, chairperson, webinar management team for giving us opportunity to conduct one-day international webinar on this very demanding topic. Now, I would like to inform that session will be closed. Again, we will meet in this platform very recently with some another demanding topic. Thank you for your support, cooperation, and active participation for the webinar and make it a grand success. So be safe. Though all you know, still I want to say in this unlock situation, please maintain social distancing, use mask, hand sanitizer, wash your hand with soap frequently, do exercise regularly, don't go outside your home except any essential work, and try to eat immune boosting functional foods as much as possible. Thank you all.
Thank you.
Now the session is over.
Thank you very much.
Thank you.
Thank
Thank
you
you,
very much.
everybody.
Thank you. Thank you.
Thank you.
Thank you very
See you.
much.
Bye.
Bye-bye.
Bye. Thank you very much.
Bye.
Thank you.
Bye. Thank you.
Niranda, please stop.
করছি
Stop YouTube.